Hello students, welcome to the Lab Activity PowerPoint for our Photosynthesis Lab. We'll be following an experiment where we've grown different plant species in different light environments, which means there are different amounts of PAR. Remember, PAR stands for photosynthetically active radiation. We want to know how these light treatments will affect the above and below ground growth of our plants. Mature plug transplants of 4 plant species were transplanted into larger containers and divided into 3 different light treatments in the greenhouse on April 3rd of this year. Plants were kept in these environments for several weeks and were watered and fertilized as needed until the experiment was taken down on May 5th. Our light environments include a shady location with the lowest PAR, an ambient location subjected to partial sun and shade, and a location with supplemental light using a sodium grow light that increases PAR. We've planted 4 different species. Begonias are known to prefer shade but can tolerate partial morning sun. It is known not to do very well in high heat or high light environments. Vinca, on the other hand, prefers full hot sun to perform best. Petunias, like vincas, prefer full sun and will have the most blooms in a full light environment. It can still tolerate some shade, though. Finally, lobelia can actually tolerate several different light environments and should do okay in all 3 treatments. It does bloom best in partial sun, but as long as it's well watered, it can tolerate full sun. Our methods are as follows. After setting up all 3 light treatments in the greenhouse, we'll use the Light Scout meter to measure photosynthetic photon flux density, which corresponds to micromoles of photons per meter squared per second of PAR. We'll visit the plants in the greenhouse after 2 to 3 weeks and take some temperature and humidity records and observe the plant growth. After 4 weeks, we'll take down the experiment, so our final measurements will include flower number, leaf number, wet shoot weight, and root density. So our specific research question is, how will these light treatments affect the above and below ground plant growth among these 4 plant species? We can formulate some hypotheses based on what we know about each species' light preferences. For instance, we know begonias prefer shade, so we would expect our leaf and flower numbers, shoot weight, and root density to be greatest in this treatment. Petunias and vincas prefer full sun, so we'd expect these measurements to be greatest in the supplemental treatment. Lobelias aren't as picky, so we might expect them to perform about the same across all treatments. Here is a picture of our experiment setup. Notice the shade environment has a tarp covering the table where the plants are being grown. This ensures less sun exposure. Our supplemental treatment has a high-pressure sodium grow light above it. These grow lights produce a red spectrum light and generate heat. They are a popular choice for horticulturalists because they're inexpensive and easy to use, as long as you fully research the ideal growing conditions of your plants. Let's confirm that our PAR is actually different across treatments. The Light Scout meter reads only 367 micromoles per meter squared per second of PAR in the shade treatment. Ambient is much higher with a reading of 1,349 micromoles per meter squared per second of PAR, and supplemental is certainly the greatest with a PAR reading of 1,981 micromoles per meter per second of PAR. After 2 weeks, we took some temperature and humidity measurements. These are influenced by PAR and vice versa. 22 degrees Celsius is about 71.6 degrees Fahrenheit. Pretty comfortable conditions for a student working in the greenhouse. Here are some images of all of the plants in this shade treatment. Petunias actually look pretty good considering they prefer full sun. Begonias are growing well too. Vincas and lobelias are still very small, but it's only been 2 weeks since transplanting. In the ambient treatment, we recorded about 24 degrees Celsius, which is 75 degrees Fahrenheit, with a 55% relative humidity reading. Again, petunias look good, and lobelias and vincas don't look too different than they did in the shade treatment. What do you think about the begonias compared with the shade treatment, though? To me, they seem quite a bit smaller. Notice the drastic difference in temperature in the supplemental light treatment compared with the other two treatments. 31 degrees Celsius is about 88 degrees Fahrenheit. This treatment also has the lowest relative humidity. It's hard to notice the blooms on the petunias because they're washed out by the orange light, but there looks to be more blooms compared with the other two treatments. The begonia seems smaller compared with the other treatments, while the lobelia and vincas, again, don't look too different. When you're performing an experiment like this, it's important to take some observations along the way. Now let's make some final observations of our plants. Here are some side-by-side photos of all the plants in each treatment. There are some sure differences. Let's look at the plants one by one. Petunias certainly have more blooms in the supplemental treatment compared with the shade treatment. They look to be a little wilty though, but we're not taking biomass measurements and so can't confirm any differences in plant production. Vincas didn't really bloom in any of the treatments. Perhaps it was the stock supply we got these plants from, or we need to modify their light preferences. This was also a very short experiment. It's important to understand when you're working with live organisms like plants, they're not always going to do what you expect them to do. To me, they do seem to be a little larger in the supplemental treatment compared with the ambient and shade treatments, though. Our begonias definitely look larger and have more blooms in the shade treatment compared with the other two treatments. This is what we expected based on what we knew about the plant species' light preferences. It's nice to support our hypothesis. Lobelias are kind of all over the place. They were our species that we expected to perform equally well across all treatments. Those were just some visual observations, and although they are incredibly helpful, we need to collect specific data to better answer our research question. Leaf and flower numbers were collected by simply counting the number of leaves and flowers on the plants in each treatment. We did grow 8 plants per treatment, but in the interest of time, we chose to take only measurements from 4 of the 8 plants. To record wet shoot weight, we separate the shoot from the roots and clean off any soil or residue before weighing the shoots. The root density was collected by cleaning the soil off the roots, first by using our hands and taking off as much as we could, then rinsing them in water to remove even more soil. In a more professional experiment, you might use a dry sieve method by using a sieve with a small mesh size to clean the roots to remove more soil, by shaking them in the sieve with a lid cover. There are also protocols to wet sieve roots to remove all of the soil from the roots. Both processes are very tedious and unnecessary for the purposes of this class. After cleaning the roots, we applied a score of between 1 and 5 to measure root density, 1 being the smallest root density and 5 the largest. This may seem arbitrary, but it's actually a very common method, sometimes used in plant pathology. Pathologists will observe disease symptoms on plants and rate them on a scale between 1 and 5. 1 being a plant showing very mild to no symptoms, and 5 is a plant that's completely dead. As long as you are consistent with assigning the scores, this is an adequate way to report the root density data. Here are the results for the petunias across all treatments. The text to the right of the tables are the average values for the 4 replicates. They indeed had the most flowers in the supplemental treatment. The other records don't seem too different from one another, although statistics would tell you for sure. Our vincas technically seem to have performed best in the supplemental treatment based on the leaf and flower numbers, but they were very small across all of the treatments. Begonias have the most blooms in the shade treatment and the highest leaf count and shoot weight. This is what we expected to see. Again, lobelias were all over the place and didn't seem to perform better or worse in any of the treatments. Here is the root density data for petunias and vincas. Remember, we applied a value of between 1 and 5 to indicate our root density scores. I took the average of these scores across the replications to get a final root density value. Petunia root density results are consistent with our other measurements and with our hypothesis. Vincas were the smallest plants across all treatments compared with all of the other species and have very low root density scores. Although begonias had greater above-ground measurements in the shade treatment, as we expected, the root density was greatest in the ambient treatment compared with the other two treatments. Lobelias had the lowest root density in the shade treatment compared with the other two treatments, but the above-ground measurements were not very exciting. Okay, let's try to summarize what we did. Although we only changed a single variable, being PAR, we inevitably changed several other factors. What other factors can you think of that might be influenced by these light treatments and therefore affect plant growth and response? You could see that the PAR did impact temperature and relative humidity. Are there any others you can think of? Go back to previous labs and think about drought stress and evapotranspiration. Did we see what we expected to see? Think about the plant light preferences. It's possible plants won't behave like they're supposed to. This is true in my own research. Sometimes you just have to accept that the data is what the data is. What if you were hired to plant one of these species in a landscape with highly variable PAR? Which would you choose? There's no wrong answer as long as your scientific reasoning is sound. Ponder this slide before attempting the quiz, and you should perform just fine.